we're going to be talking about cell division, what it is, why it should really be called nuclear division, and the processes by which different cells divide. So stomatic versus reproductive. So to begin, mitosis is the first process we're going to be looking at. It's pretty much the basis for all cell division, just because once you understand mitosis, you'll be able to understand meiosis, which is a little more complicated version for different cells. So for stomatic cells, which are every cell in your body except the reproductive cells. So for example, skin cells, muscle cells, heart cells, brain cells, and all that jazz would be stomatic cells. They divide through mitosis, where the parent cell divides into two identical daughter cells. They're identical to each other and to the parent cell. And we'll see why it's like this. So the phase right before mitosis is when the cell is growing and synthesizing its DNA and centrosomes. So as you can see, there are two centrosomes that start migrating to opposite sides. This is going to come into a later part of mitosis, but know that there are two there in interphase. They start replicating there. The DNA is not in condensed chromosome form, which we're going to be looking at through mitosis. It's in chromatin. It's very unraveled, and if you look at it through a microscope, you'll just see a solid black circle or ellipse or whatever shape it is, because you can't make out the distinct chromatin. It's located in the nuclear membrane, and there's a cell membrane. So the centrosomes are in the cytoplasm of the cell. So going to the first stage of mitosis, it's prophase. So prophase is where the nuclear membrane starts fragmenting, which is when it starts dissolving. The chromatin starts condensing into chromosomes and starts to join with its identical pair of DNA to form sister chromatids. And the centrosomes are almost at their asters, or the opposite sides of the cell. You can call them poles. So once we move to metaphase, which is where there's no more nuclear membrane, the sister chromatids line up at the metaphase plate, which is an imaginary halfway point, like equator between the cell. Spindle fibers, which are microtubules, start connecting to the sister chromatids on each side to pull them apart in the next phase. So to pull them to each aster. And the point of lining these chromatids up on the plate is to make sure that one cell, when divided into two, does not contain two identical DNA sets. So each cell wants one set, but a cell does not want two sets, because that means the other cell will not have a set and will be missing DNA. So this is to avoid that. And so in anaphase, the sister chromatids are separated by the kinetochore microtubules, which are the ones that are attached to the chromatids, or not chromosomes, shortening. So they start shortening and pulling apart these sister chromatids to each side. And then after this comes telophase. So telophase is where the cell starts to pinch. This is where the nuclear membrane starts to form again around the DNA that was taken to each aster. And the chromosomes inside actually start unraveling, where they start going back into chromatin form. And if you notice, the cleavage furrow is where it's pinching the cell. But the centrosomes, now there's one in each cell. You need two to divide. So in interphase, that's where another centrosome is created. And that way you can have one go to each aster or each pole of the cell to pull them apart again, and to divide into two more cells. So in telophase, the DNA is now split into two. Now they're the same exact amount of chromosomes, 23, in animal cells, as the parent cell. So this way, these cells are identical once created through cytokinesis, where the cells actually split. They're identical to the parent cell and to each other. Now, what's different in meiosis, which is the other process, is that in meiosis, the results, which are gametes at the end, will be different than the parent cells and the daughter cells, because the gametes only contain half the amount of DNA as the parent cells have, but the same amount of chromosomes. You'll see why. So reproductive cells are the egg and sperm cells. That's what join to form you, or to form the zygote that you come from. So let's look at the first part of meiosis, which is meiosis 1. So in this part, there's prophase 1, which is similar to prophase, except the chromosomes start to cross over. So the crossing over does not happen between sister chromatids, but between homologous sister chromatids, or homologous chromosomes. So that's when DNA from one part goes to DNA of the other. And that way it creates variation, because when you look at people with multiple children, they're not the exact same. They all have their differences that come from each parent. And this allows there to be variation. Crossing over mixes up the DNA. So each sister chromatid is attached at the centromere, and the nuclear membrane is fragmenting, and the centrosomes are moving to the asters. So after prophase 1 comes metaphase 1. Metaphase 1 is the same as metaphase in mitosis, except that now they attach to sister chromatids on each side, and they line up as homologous chromosomes. So this way, each cell will get 2 pairs of DNA rather than 1, because after meiosis 1 there's another split, which is meiosis 2. But anyway, the kinetic core microtubules connect to each chromatid. The non-kinetic microtubules do not connect. They are they're not relevant to shortening the chromatids. And in anaphase 1, the centrosomes will pull apart these chromatids. So now each cell, the homologous chromosomes separate, and each cell will now have 2 sets of DNA. Telophase is where it starts to form the nuclear membrane, and 2 cells are formed again. But if you notice, they have the same amount of chromosomes as we began with, so 23 into 23. And now, after telophase 1, after they're 2 distinct cells, they're not identical to each other, they're not identical to the parent cell. They have the same amount of DNA as the parent cell, but they're not identical. After these 2 cells form, there's another centrosome that replicates in both, so that these cells can divide again. So this is in meiosis 2, which are where gametes are formed. And gametes are the resultants of meiosis. They're what form the zygote, where they combine the egg and the sperm. So we're going to show both cells that resulted from the previous phase, meiosis 1, telophase 1, you can say, to show the differences, or really how they're similar, but they result in different gametes. So again, there are 2 centrosomes, the DNA condenses, sister chromatids are formed. And now, besides the DNA not being the same, or the chromatids that form the sister chromatids being the same in both, it follows the same path as mitosis. So they line up at the metaphase plate, where as sister chromatids, the spindle fibers attach, and in phase 2, they're pulled apart. So if you notice, the DNA is different because of the crossing over in each side. So top of the left one, bottom of the left one, top of the right one, bottom of the right one. None of them are the same. Telophase is where you can see it. So the amount of chromosomes in each of these gametes, or the resultants of meiosis 2, are 23. It's similar to the parent cell. It's similar to the daughter cells that result from the parent cells, or the resultants of meiosis 1, but the DNA is halved. So there still are 23 chromosomes, but once they unravel and aren't condensed, there is half the amount of DNA. Because what happens is, one gamete combines with another gamete from another person to create a child, or to create a zygote. That way they still have 23 chromosomes, and instead of DNA from each parent. So these cells have a specific purpose. They're to reproduce only. And you can see how they really differ from mitosis, but they follow that same pattern of lining up, condensing, lining up, splitting, and then the cell splitting. So if you see, mitosis and meiosis are really just the splitting of the DNA, to have each DNA go into two separate nuclei, while cytokinesis is where the cell actually splits. So it happens after mitosis, it happens after meiosis 1 and meiosis 2. And that's how the different distinct cells are formed, rather than just the nuclei being formed. So that's pretty much it. Thank you.